Good afternoon, everyone. Thank you so much for joining this webinar today. This is episode 7 of the Community Connect webinar series. The aim of this webinar series is for us to give people from the community an opportunity to come and tell us and teach us and provide us with tools and information and sometimes opportunities that we can make use of. So today, that in mind, we've got Rensu Theart here. Rensu is a senior lecturer at the University of Stellenbosch in the Department of Electrical and Electronic Engineering. Rensu's primary research is in the field of image analysis and machine learning. During his PhD, work, he specialized in biological image analysis and visualization systems, including the development of virtual reality tools. He has since then widened his focus to include facial recognition and markerless human pose estimation, among other things. These are very, very interesting initiatives, and are initiatives that I would encourage you all to look into or learn about. Very important work. So I think he's the perfect candidate to come and tell us about AI and bioimaging processing and the different applications and techniques that we can use it with. So with that, and no further ado, Rensu.
So thanks everyone for joining. I will be talking about some of the work that I've been involved with, and in the process, my aim with this meeting is to kind of teach you or show you how I was thinking through these problems as I was trying to solve them on a quite a high level. Because I'm not exactly sure where everybody's expertise are, and in terms of image analysis and machine learning. So there might be some foundational things that I'm going to be telling you, and I hope I can still make it interesting enough that even if you've heard it, that you will still learn something new from it. But yeah, with that, let's jump in. So I'm just going to start by explaining a little bit about digital images and image processing. So the idea of image processing is that you take these digital images and you put them through some algorithm to give you some information out of these images that you wouldn't otherwise have from just visually looking at them. And the source of these images can be from many places. It can be from cameras, can be from medical imaging devices such as MRIs or CT scans, confocal microscopes, satellites, whatever you can think of. But what they all have in common is that there is some kind of an image sensor which contains light-sensitive elements, which then capture basically the intensity of light that hits that region of the sensor. And you can put that image sensor in a camera, or you could put it in a confocal microscope or wherever you need it. And then ultimately what you get out of this is from the sensor is something that we visually interpret as an image. But the reality is that this image doesn't really exist. Internally, for example, this photo would be made up of 3 components, a red, green, and blue component. And you can kind of see what would be represented in these components. For example, the yellow is represented by a combination of red and green light, but there's no blue light, for example, there. And white would be a combination of all 3. You can also look at this red flower, which has this yellow border in the red tone. It looks like a consistent color across, but in the green you can see that there's this fringe. So you start to understand that, oh, actually there are different components that are represented behind it, but still we're not actually having an image. What we're really working with is just a matrix of values. And if you zoom into an image in some software and you pay attention to the numbers on the side here, as I move across the pixels, you can basically see the intensity values that are sitting behind those what we perceive as colors, but actually it's just a matrix of 3 channels of values. Now, sorry, there's someone's microphone that's on. So yeah, given that this is now how something that we call an image really exists, it's not something that's actually got some structure necessarily as a visual perception thing, but actually it's just a matrix of values that's sitting behind it. We must use this reality of images to now put them through some algorithms to ultimately produce some interesting analysis results. So the first thing I'm going to talk about is mitochondria, and me being an engineer, having a little bit of background from biology from school, this was the general understanding that I had of mitochondria. It's the powerhouse of the cell. There are basically, as you can see, 4 mitochondria in the cell, and they are just little blobs that float around, not interacting with anything, So this is the picture that many people, often just not having necessarily a deep understanding of biology, have the perception of what's going on in a cell. But the reality is a whole lot more complicated than that. So these are images captured under a confocal microscope where we stained only the mitochondria. So everything else is black in the image. There are two cells. You can kind of make out the nuclei here in the middle, but then everything that's white is mitochondria. And you can see that they fill basically the majority of the cell. They're mostly networked close to the nucleus, but they are very highly dynamic. They're remodeling themselves, building networked structures within the cell, constantly remodeling itself as as the cell undergoes various stressors or metabolic needs. It changes its structure. And what is going on here is that we expose the cells to CCCP, so they're basically busy dying. And you can see in the response to try to stay alive, they are fusing together. The network is becoming very dense to respond to the stressor. So we wanted to model this, and there are a few reasons why, but just to explain what we wanted to achieve is we wanted to find in this network, where are there certain mitochondrial structures which would break out apart into two separate structures, or where are there two structures that would join together? And this obviously happens throughout the entire mitochondrial network. And then the third one, which I'm not going to focus on too much, is where are the mitochondria that then lose their membrane potential, which means that they're basically will be degraded soon, and they're not no longer useful for the cell. And what we actually wanted to have is we wanted to have some algorithm or some system where we can put our image in that we captured very nice and beautiful mitochondrial image. And then we wanted the system to tell us where will these fission, fusion, and depolarization events occur. Now, there are a few things why this is important, because if we see that there's generally a higher rate of fusion than fission, for example, it means that the cell is becoming more condensed. If fission is higher than fusion, it means the cell is busy spreading out or breaking apart. And these have some implications in terms of the mitochondrial dynamics and the general health and the viability of the cell. So if we can model this, we have a nice insight into what's actually going on in the mitochondria. And mitochondria has been shown to be very central to various diseases. Like if there's mitochondrial dysfunction, it often results in other things such as neurodegenerative diseases, such as Alzheimer's disease, Parkinson's, can also lead to brain cancer, for example, gliomas. So if the mitochondria starts to go wrong, then a lot of other things go wrong. And in the process of trying to find drugs to treat these types of conditions, we want to have a very good way of understanding how the mitochondria is behaving in this current cell under various treatment conditions. So I've really kind of said that. And one of the goals here is that we wanted to know the number of events, fission, fusion, depolarization, but we also wanted to know the exact location where they occur. Where, for example, if there is, you would expect there to be more fusion and fission at the edge of the cell, but a lot less closer to the nucleus. So just knowing where it happens is also kind of insightful for mitochondrial dynamics. But remember, behind everything, behind this thing that we perceive as an image, there is actually only a matrix of values. There is no mitochondria. There is no structure. Really, we just have intensity values. And then from the intensity values, we must make some judgment call as to where these events will now occur. Now, the traditional approach what we would have for other more simple image processing tasks is you have your input image, you have some determined algorithm, and then that produces some output image. And you can think of this as kind of a mathematical function in some sense. So if you know a little bit of Python, this is like a Python way of writing code. We have some input into some function. This input, in this case, is degrees Celsius being converted to degrees Fahrenheit. This input, there's some well-defined formula that says, okay, well, you need to multiply the degrees Celsius with 1.8 plus 32, then you get degrees Fahrenheit. And yeah, this is a very deterministic system. We know what output we will be getting given that we know this formula. The problem in our case is we don't know what this algorithm is. There is no defined algorithm that we can map the input to the output. So in a situation like that, what we usually resort to is machine learning approaches. And how machine learning works, just as a nutshell, is that you have some input-output pairs, in this case now, and then you have a neural network or some other machine learning model that forms the base of your algorithm. But initially, it's randomly initialized. There's no prior knowledge that you put into it. It's just, we have no idea how it maps, so we just randomly initialize it. And then through some process of training, we then learn what this algorithm is, so that in future we can give a new example as an input, and then the output that will be produced would be sensible. So in short, machine learning is you give some input data, you give some output data, you put that into some machine learning system, and you get a rule or an algorithm out. That's kind of how you should think about machine learning. And what we would typically want to apply in this context is a subfield of machine learning known as deep learning, where we use deep neural networks, which add multiple layers in these deep mathematically connected networks that are able to, they're called universal function approximators. They can approximate almost any function, any mapping from one domain to another domain, if you just set this thing up properly. And the way that these deep neural networks are designed is it's inspired by how nerve cells in a human or a biological brain are connected, and with the synapses and everything. That's kind of the foundation of what inspired these deep neural networks. Okay, now a very brief nutshell of how neural network training works is you have a forward pass. You initially have your network here. So this is the input. You give it to your network. This is the network that's initially entirely randomly set up. There's got no structure related to it. So then you put that through this mathematical function, and then you predict a value from that. Now, initially, this prediction will be absolute rubbish. It will have no resemblance of anything close to what you want. But this is the first step. You have an input value, you make some prediction from it, given your randomly initialized network. We call this the forward pass. And then obviously that value will be bad initially. So we calculate what we call the loss. And the loss is basically, in a simple sense, what is the difference between what I expected to get and what was the predicted value. And then I calculate what this difference is between the expected value and the predicted value. And then through some optimization process, which we then call the backpropagation or the backwards pass, we then update these layer values so that the next time we try to run this to make a prediction, it will make a much better prediction. And then we repeat this process: forward pass, make a prediction; backward pass, update the weights, and then ultimately the network learns to approximate any function. And one of the key steps of ways that you optimize these parameters is you do something called gradient descent, where you have your loss function, where this is the expected value, the predicted value, how far they're apart. And then we say, okay, well, in which direction do we need to update our weights so that in the future they produce a better, closer to the reality output. And you step down this gradient until you get to your optimal location. Now, this is only for one weight in your entire network, one parameter, if you will, in your network. And it gets complicated quite quickly. So if you just go to two parameters in your network, then this is what the loss function could, for example, look like. You initially have something randomly initialized. You have a point that's generated that's quite bad. It's got a very high loss. And then you try to walk down this loss curve to ultimately get to a neural network that produces the best value. And you can see if I started walking in this direction, I would have gotten stuck there. I couldn't walk further down there. So then I haven't really fully optimized the network. So that's one of the challenges of neural network training, is to find a way to get to the absolute optimal point in your entire landscape. And this is just two parameters. Usually we work with millions or sometimes billions of parameters. I think in the newest large language models, they use trillions of parameters that needs to be optimized together in this way. So it can get quite challenging. Just a quick introduction to convolutional neural networks. So now that we can train neural networks, we have an idea. We don't want to just put our image in its entirety in. We want to say that some structural information should be retained in this prediction. And that's where convolutional neural networks come onto the scene. They the principle of how they work is you've got this little kernel for which you learn the parameters. This is the only part that you learn. And then you just slide this kernel across your image. And then as you get to a certain point, you just simply multiply the values together. So the top corners get multiplied together. Then the next one are multiplied together. You do this for all of the cells, and then you just sum it up. And then that sum is what you then put in your convoluted image at the end of the day. You do this for your entire image. On the edges, you would typically pad it so that you keep the image size the same. And then you have a convoluted image that is now some representation of the previous layer. And why we want to do this is we want to transform the features in our image to something that ultimately is of a more lower dimension state, so that we can use this low dimensional state to make a prediction for whatever we want to achieve. And I'll get a little bit back to that now. How we get to that lower dimensional state is often through something called max pooling, where we take our convoluted image and we simply say, in a little square, what is the maximum value? We just take that maximum value and we retain that. We throw everything else away. There we retain the 30 and etc. So now we've just made our image a little bit smaller. And what we achieve by doing this is, if you think about a face, initially you would have just very high detailed features that you would look at, like what are the edges? Is it the edge from the top, the side, or whatever? You just look edges or little curves. And then as you max spool, it's as if you're conceptually looking at a broader part of the image at a time. So now you start to train the neural network to identify eyes or a mouth, for example. And then at the very last layers, you can start identifying an entire face in terms of how you've now restricted this. So this is a very common CNN architecture where you have, let's say, a handwritten digit. This is not just an example. You have these convolutional layers. You bring it down dimensionality. You flatten it. You get your a fully connected layer, and ultimately you have, in this case now, 10 categories, which it must then select which one is the most likely category that is associated to this image that I put in. In this case, it obviously be 2. Okay, now this is just generally processing images, but what we want to do, I can just remind you of our objective, is I want to take an input image and I want to generate another image which shows me where these dots occur. And for this, we usually resort to image generators such as a generative adversarial network. I'm not going to go into the detail of the math behind it, but I'll explain this in a kind of analogy way. So we have the one neural network, which is the generator. This generator is tasked with generating images that look like real images, the real ground truth that we're trying to get to, but actually it's obviously generated. So it's all that it's doing, it's kind of making fake money. Now there is a real image in our case, which is uses reference to compare against, and then we train another neural network to be able to identify when something is fake or real. So what happens in this process is if our discriminator neural network becomes very good at identifying what is real and what is fake, it starts to say, I know what to look at in order to tell you whether something is real or fake. So in this case, if you look at this picture, there's a little mustache in this picture here. And the discriminator says, I see a mustache. Okay, therefore, this is a fake image. So then it can give feedback to the generator to say, hey, when you do this thing, I can very easily identify that it's a fake image. So stop doing this thing. Make it a little bit more like real money. And if you train them against each other in this way, the discriminator gets better and better at predicting or telling you which is fake, and the generator gets better at generating something that's indistinguishable from the real thing. So that is what we want to do: input image, output image. The problem with our general GAN is that it doesn't take an image in; it actually takes some random noise in. So we use a different type of GAN called a conditional GAN, where you add this additional label information, which gives you the context for which you want to train against. And it's kind of helpful to illustrate it in this way, where you give a hand-drawn sketch, and it's supposed to predict an actual shoe. Like it's supposed to generate a picture, a photorealistic picture of a shoe. And then when we give it to the discriminator, we give this hand-drawn picture as well as this predicted generated image. And then it's based on these two that is trying to tell you whether it's fake or real. And if it was real, it would have received also these two. But now, because we've got some labeled input, we have structure to inform what our generator should be generating according to, which is obviously what we need, because we don't want to generate some random mitochondria. We want to generate these dots on top of our existing mitochondria. And the neural network that's sitting behind this generator that achieves all of this is a unit. In case you've heard about this, the basic structure of it is that you bring the dimensionality down to some bottleneck, and then you raise it up again to get the full image resolution that you had at the input. And you have these skip connections to give the network a little bit of help as to retaining the understanding of the structural information from the high resolution image all the way up until the final output, while forcing it to go through this bottleneck to actually extract the main understanding of the features of your image. And when this was initially published by the people that developed this method, they demonstrated that it can work in quite a lot of domains. It can be from hand-drawn images, it could be from these segmented facades to actually generating photorealistic images from daytime to nighttime, etc. So this is the perfect type of architecture that we want to use. Now, there's been expansion on this called voxel to voxel or vox to vox, which works on three-dimensional images, which is what we actually typically have from a microscope, is these Z stacks of confocal images. So now if we look at this picture, previously I said we don't know the algorithm, but now we know what the algorithm is. It's a neural network, and it can be this conditional GAN, pix-to-pix, vox-to-vox, this type of networks I've been talking about. But now the big problem is, to train these things, we need to give it the input and the output. We need to have the ground truth of where these dots are actually at in order to train the neural network to produce the fake image of where it really is at, or where we think it's going to be at. So we still have the same problem as to, we don't actually have this ground truth, which is the thing where we're trying to generate in the first place. But the good thing is that we can actually generate this by hand, or not by hand, but develop a method that can generate this for us. And the way we went about it is we said, okay, well, if we capture a time-lapse sequence of this, the cell or movie of the cell, of a live cell that's moving around, all still in 3D and Z stacks, we can then extract 2 consecutive frames from this video. And then through some processing, which I'll talk a little bit more about now, we can then determine what is the difference between these two images. Were there two structures that became one structure, or where was there one structure that became two structures? And then through this back and forth comparison, we can ultimately then generate these dots. So then we have this input-output pair that we would need for the neural network. And just a little bit of detail as to how the initial version of this method looked. It's been a little bit adapted since then, but we start by deconvolution. I'll talk more about that just now. Then we upscale the image, and the reason for upscaling is we want to do a segmentation at the end of the day. And the segmentation or the binarization is really important, that structures that are close together that should be separate will actually be separate. And if the resolution is too low, sometimes you in your binarization, you've got two pixels that are touching each other, but actually they're not supposed to be touching each other. So if you upscale the image a little bit before you actually do the binarization, you force these structures just a little bit further apart so that when the binarization happens, it doesn't join them. Gaussian blurring just to get rid of some noise, contrast stretching to get images a bit more consistent in their histogram intensities. And then we used a method called hysteresis thresholding. You can use any way to threshold images for this method. It's not dependent on this, but the benefit of hysteresis thresholding is in this goal of keeping things separate that should be separate and keeping things joined that should be joined, what hysteresis thresholding does well is it says, I've got two thresholds. If I have a pixel that has an intensity above the highest threshold, then it's definitely binarized, it's foreground, I want to keep it. If something is below the low threshold, then definitely throw it away. It's definitely background noise. But everything in between, there's a little bit of uncertainty. So what hysteresis thresholding then does is it says, if I've got an intensity that is between these two thresholds and it's touching a structure that has already been binarized as being a foreground, then I'm going to add this to be foreground. But if it's touching something that is considered to be background, then I'm going to keep throwing it away, that it doesn't keep growing the structures out. And that works quite well for this type of application. So I'm going to not talk about the rest of the steps just now. I'm just going to show you briefly that there were a lot of mathematical back and forth comparisons that ultimately led to the output. Comment on deconvolution. If you are not aware, deconvolution is a step that's necessary for microscopy, where the lens itself causes some refractive artifacts in your image. But this is, you can model this mathematically. So this blurring that's introduced by the lens, the optical properties of your microscope can be removed. And you can see that if I was to work with this left-hand side image, it's much more clear after I've removed that blurring that I could model. And this process is called deconvolution. And now I can actually make a cleaner distinction as what is touching and what is not, for example. And yeah, just a point on the challenge of binarization. So with binarization, it's not that you keep constantly fighting two competing objectives. So for example, if you consider the structure there, they are clearly two separate things. They should not be binarized as being joined together. But on the other hand, if I've got this structure here, you can see there's this dip in intensity. And I actually want to have this as one continuous structure after the binarization, because it is, from my eye, I can clearly see this is one continuous structure. Now, if you don't necessarily do this trade-off properly, you can end up with something like this. So you, after the binarization, now these two, which should have been separate, has now clearly been joined together, which is not correct. But that structure, just to show you again, that structure there on the left, which had this dip in intensity, is now nicely joined. So there's often this trade-off, and it's very difficult to get one method that just works for everything. So often you have to say, okay, well, I'm going to just accept the fact that it's not perfect, and I'm going to make sure that my comparisons are consistent, and then that's kind of the best you can do in some cases. Neural network segmentation or binarization is definitely making this much better to deal with these types of nuances, but any deterministic methods tend to struggle with these types of applications. Okay, so after we've now done this, just some results. So this is an example from a control cell, which is supposed to be a healthy cell that is just happily going along its way. And you can see the fission and fusion rates are nice in equilibrium. So the cell isn't breaking apart. It's not massively shrinking in or joining together. It's there's a nice equilibrium between fission and fusion events. And just to maybe, show you a little bit more an example here. So you can see, for example, this one, initially the structures are separated, and then in the next frame, this is now frame 1 and frame 2. You can see in frame 2 they are nicely joined together, so that would clearly be a fusion event. Or on this side here, it's running a little bit off the frame, but it's one continuous structure. And then you can see in this side, it's now clearly being separated as two structures. And then you can see this fusion event there. Just another nice one is that there's this phenomenon that was described in literature since quite a while ago, which is known as kiss and run. We've got two mitochondrial structures coming together. They briefly fuse, they stay fused for a bit, and then they just break apart. And we can now automatically detect these types of events with this method, because we just look at a certain window in our entire image and say, well, are there any events that over a sequence of time alternate between fission and fusion? And what you can see here is you've got two structures initially separate, then they fuse, then they stay fused for a little bit, and then they again break apart or they undergo fission, and that's indicated by this red event there. So yeah, this has been quite a helpful tool in our toolbox to give us more information about mitochondria. I just want to show you some shortcomings of the initial implementation. Most of these have been solved in the latter implementations, but this is, I'm just showing you a little bit behind the scenes, because I think this is good to, if you develop methods like this, that you think about some of these assumptions that might be made. So one of the big challenges, as I was saying, is with the binarization. So let's take this one. This is a nice clean one. When you've got this structure here, I want to have that binarized to be one continuous structure. But in the process, there is a little low intensity bridge there between these two structures, which are clearly separate. And if I don't binarize properly, these are going to be joined together as one continuous structure. And then in the next frame, where they just slightly moved a little bit further apart, then suddenly that low intensity bridge is not as prominent. And now it's binarized as two separate structures. So then this would have, by this method, been incorrectly labeled as a fission event, which means they're breaking apart, which in reality never happened, just because the binarization was a challenge. Another challenge here was, initially, when I developed the method, I was not really sure where to indicate the actual location of the event. So I said, okay, well, where is it most likely to happen? Well, if I've got two structures that join together, then clearly the point that's closest between the two of them would be the most likely location of joining. So for example, if it would be that point there between the two structures, that would be where it joins. But this is where it goes wrong. If you've got, you can now see the red and the blue, sorry, the red and the green, they are the two structures in frame one, and then the gray is what the new structure looked like in frame two. And you can see initially these two structures are not joined, and then in the next frame, they both simultaneously joined to a third structure here on the side. And now my algorithm initially just said, okay, well, let's just find the closest point between the two structures. And then I said, okay, well, the closest point between these two is simply that green dot there. So that's the location of fusion, which is obviously not true. There are, in fact, two locations of fusion there, and they have joined to a third structure. So now what I'm doing is, instead of just naively taking the closest point, I now skeletonize this entire structure, this binarized structure in 3D. And then I try to follow the path from the one structure to the other structure. So I'm saying, initially I'm in this structure. Can you find a route to get to this structure? And then wherever you move through different structures, then mark that as fusion events. So now it's going to run through here, mark that as a fusion event, run through, mark that as another fusion event, and then you have the correct location for your events indicated. And this improved version has been wrapped up in a Fuji plugin. You're welcome to use it. I would suggest that you contact me if you are interested in using it. It's easy to use, but it might take a little bit of insight to just get going. It's not super user friendly at the moment, but that's always on my radar to try to improve in the future. Okay, now I've been talking a lot about the method, how we generated the ground truth, but let's now actually get back to what we were talking about initially. I've got the ground truth now. Now I want to actually predict the output. So we did this. We trained a neural network on these input-output pairs, and this is now what our validation loss looks like. You can see we're following down this loss curve, and then once it starts going up, that means you're starting to memorize your training data, or you're overfitting is the word that we use, and that's the place where we want to stop. So stop the neural network there, and this was some of the preliminary results. Now, we did multiple experiments where we gave both frames in, and then it actually produced pretty good results. But I want to highlight this frame because this is an interesting one for me, where we only gave in a single frame. It's a Z stack, but it's only a single time point. We didn't give any video or time context for this neural network. And then it predicted all of these events. Now, some of them are very easy to filter out. So for example, low intensity outputs, we can easily just filter them out. We just disregard them. Another thing which we also decided to disregard before we did the validation is where there are two events predicted at the same location. So you can see here, there's not a lot going on there. So the neural network thought maybe there could be something there, but it's not sure if it's going to be fission or fusion. So then it just predicted both. And then fission, it's red and green. So it marked it as yellow. And in this uncertain case, usually that is just erroneous, and we can also disregard that. But then the events that remained, we validated them, and there was about a 50% accuracy, meaning that when the neural net predicted there is an event, then we looked at the actual images before and after, and then we found, yes, there is an event, or no, there is not an event, and about 50% of them was accurate. Now, what's really interesting about that result is, well, first of all, not that it's really usable for anything practical, but that means that what is going to happen within the next 10 seconds of this cell is already written in the morphology of the structure of the mitochondria, so that the morphological structure of the mitochondria is giving a very strong indication as to where these events are likely to happen. So that gives actually quite a good amount of hope that if you were to analyze the morphology, then you would have a lot of insight as to the longevity of the cell. So this is now something that we also did, and this is now a bit of a different application, but we extracted a lot of these morphological parameters. So we took our binarized image in 3D, and then we extracted lots of parameters such as volume, surface area, form factor, aspect ratio, solidity, sphericity, eccentricity, a lot of them. I think there were about 30 in the end of the day of all of the different things that we looked at. And then we say, okay, well, if we have different treatment groups and we push this through some dimensionality reduction method such as PCA or LDA, or we push them through some machine learning type of methods such as t-SNE or UMAP, what do we see? And I'm just going to show you the results from the LDA, because the LDA is a deterministic method where it looks at different clusters and tries to find the angle to look at these clusters to give the best separation between them. And this is what the LDA looked like of this experiment, where we had control cells that were insulin resistant, and then fatty acid treated cells, and then other cells which were both insulin resistant and had fatty acid treatment. And I'm not a biologist, I'm an engineer, but what the hypothesis was that that this fatty acid and insulin resistant cells would, morphologically speaking, look very similar to just the fatty acid treated cells, but different from the other two. And after just extracting these morphological parameters of the mitochondria, you could see these very clear clusters forming. And also this LDA method was not able to distinguish between these two clusters, which was exactly as was hypothesized. So that's kind of one of the benefits of just looking at the morphology of the mitochondria, is it tells you a lot of what is actually going on in the health of the cell, of the viability of the cell, where it's heading, etc. So I've got a couple of more minutes. I'm not going to go too much in depth on this, but I wanted to just demonstrate that a very different application is building a method that for correlation, colocalization analysis, where we qualitatively visualize what the correlation is in the colocalization, instead of just trying to look at the overlap between them or just trying to look at the Pearson's correlation coefficient. We actually can see the visual intensity of these colocalized regions, which gives us a much better nuanced understanding of where colocalization happens. But you can ask me more about that if you want to. We also bought this image processing pipeline where you can you've got all of these nodes available to you, where you can connect them in this sequence, this pipeline way. And then as you change a parameter in an early stage of the pipeline, you can see those changes reflect all the way downstream in the pipeline. Then you can also build multiple pipelines where you're not sure which one of these parameters are necessarily the best. Then you can just all visually see this happening in real time in front of you as you change some of the parameters. In the earlier stages, as I was saying, you can see those changes reflecting all of these things. And especially when you want to do binarization, as I was emphasizing previously, you want to be very sure that your binarized result is good. But it's very tedious to change the parameter and then run a Fiji macro or whatever, and then see what the images look like. It's quite a long process. So this is just more interactive to just create your pipeline. And then we also at the moment have some morphological parameter calculations from this. But my general suggestion at this stage would be, if you were to use this pipeline, then you should rather repackage it in a Fiji macro, for example, to do batch processing. But this system can do batch processing and general analysis. And then just as a very last demonstration, this was the virtual reality system that Precious mentioned that I was working on during my PhD, where we take our confocus samples, visualize them in virtual reality 3D, which gives you a much better intuition as to what you're actually looking at, especially if you want to make region of interest selections. You can just lean around the thing in 3D and then see what the actual selection looks like. And here, for example, I'm just going to go back a little bit. We wanted to make some selections of the perinuclear region. You can put one selection outside of the nucleus, one selection inside, and again, very easy to validate that in VR and extract that region to do analysis specifically only in that region. And the white part that you're looking at is localized voxels that we're just highlighting to see what we're looking at. We then expanded this idea of a virtuality system to a teaching tool, where we then put we aimed it at second year physiology students, where we explained to them the different structures of the cell with real world samples. Because as I was saying initially, my perception of mitochondria was only these few things floating around. And I think a lot of biology students even have that perception until they get to postgrad level. And this system was designed to bridge that gap a little bit. So you can now pick up the nucleus, see what it looks like. You talk about mitochondria, and then, there now you can pick up the mitochondria. You can see that it's filling the entire space of the cell. Then we immediately show them images of, again, this is in 3D. We don't fully appreciate it on the screen, but these cells are 3D in front of the user. And then you can see these fission and fusion events happening in this time-lapse sequence. So a lot of intuition that's just added by putting someone through this experience instead of just explaining something from science or a textbook. And yeah, just as a final thing, here's the autophagy process shown with an autophagosome coming around some cargo, and and then a lysosome coming in, fusing to it, and then creating an autolysosome, and yeah, then releasing everything back. But yeah, that's the presentation. Oopsie, sorry, I closed it already, but that's the presentation. Yeah, if you've got any questions, I'd be happy to answer some.
Thank you for the presentation, Rensu. It was wonderful and very informative. Please do post your questions in the chat. Otherwise, if you'd like to unmute, just raise your hand and we can unmute you. I know that Emmanuel had a question. Emmanuel, do you want to ask it yourself, or can I read it out? I'll read it out. Dear Rensu, I've entered in the CGAN PIX2PIX field since months when you stopped training. Do you base it only on the losses of the generator and descriptor, or would you base it also on your goals? For example, how good would it recognize mitochondria events?
Yeah, that's a very good question. So generally, when you train a neural network, you want to get it as close as possible to your final objective, which is in our case to predict mitochondrial events. We didn't actually end up looking at that. What was quite a challenging thing to get the thing to do in the first place is just to actually predict or generate an image which has these small dots that it actually predicts. Because as you would have seen from my other examples, the PIX2PIX usually likes to generate something that's on the full image. So it takes an image that is fully, let's say, in grayscale and then makes it fully in color or something like that. So just having our loss function defined in such a way that it really makes sure that when it gets a dot right or wrong, that it's penalized for getting that dot right or wrong, was already a challenge in itself. Yeah, we never actually went a step further and said, okay, well, are the events that are detected automatically determined to be correct or not? But that would be a very sensible thing to do, yeah, as part of neural network training.
Okay, thank you.
Rensu, in your initial stages of trying to work through this system, what did you find your biggest hurdle was? The biggest hurdle, yes, because of maybe the information, you had lack of information, or you needed more context in order for you to understand it, and the information that you then gained from experience, kind of like a troubleshooting that you would give to anyone who's starting off on this.
Yes, so there were actually a number of challenges. I think one of the main challenges I had was that I don't come from a biology field, so I wasn't always fully aware of what I was looking at or what I was supposed to be seeing in the images. So that over spending years at looking at these images and really nitpicking the results of these analysis tools, you get to learn how to interpret certain things and what to look at. So I think if anyone's just starting with image analysis, I think a big first step would just be to just play around with images, throw them through different processing, see what happens, just get a bit of a feel for how different processing methods changes the image and what you're seeing. Then in that process, trying to see, okay, well, what is the thing that I'm trying to emphasize? What is the thing that I'm trying to extract from this image? And then does this one step get me one step closer to getting that information extracted or not? So a very trial and error approach is kind of the way that I learned. Another thing that was quite a challenge is that in developing that mitochondrial event localizer where the events are detected, I was trying to emphasize this now, but you constantly had to fight against what stage in my pipeline is the thing that's not working properly. Is it the pre-processing that's not ideal that causes bad segmentation? Is it that something in the logic of my code that's supposed to detect what the difference is between one frame to the next, is the percentage overlap that I considered when considering whether one structure in one frame is associated to another structure. Like on what basis should structures be associated between frames? So there were just so many questions which nobody could really guide me, because you're developing this method from nothing. So just really struggling through all of those aspects. And I think it's just the persistence of or I suppose the encouragement from the biologist to say that, hey, listen, this would actually be very helpful for us, or it looks like it's starting to do something, just keep going and then it will be better. I think you need those voices in your life to push through those hurdles.
Exactly. And so do you think from I'm sure from an engineering perspective, there were skills that you had that kind of help bridge any gap that you have. Do you anticipate that there will be any big gaps that a biologist needs to jump in order to kind of use these tools?
Yes. So what I have realized just in general having helped some other biologists with some image processing tasks is that for me the biggest gap is understanding what I'm looking at in terms of the biology. I'm seeing some pixels moving around, but I don't know what they are. And for the biologists, it's pretty obvious. It's like, I look at the pretty pictures. They're very interesting. And for me, what comes very naturally is writing code and software, and it's second nature to me. Where for a biologist, it's not always a second nature. So that thing I was just mentioning about trying various pipelines, trying various methods, applying them, seeing how it affects the sample. The best way to iterate that is by writing code. If you want to click on all of the buttons in Fiji, you're welcome to do that, but it's going to take a long time. Writing quick macros or Python scripts that generates a whole bunch of these potential combinations of parameters, and then you can just scroll through them quickly. It's quite a valuable skill just to be able to be productive in doing this. So I think the biggest gap for someone coming from the biology field would be to get some competence in coding, and not just say, okay, I can read code, but actually can you write a little bit of code? And these days, ChatGPT and these tools are really great with just helping you get going with that. But yeah, the coding ability and actually getting comfortable with code and changing code and tweaking things and seeing what happens, I think that's the biggest thing that might not come as naturally.
Thank you. Thank you for that. We do have a question from Dr. C. Fenter. Thank you for the presentation. Is the software that helps with the steps of the image analysis, as shown, I'm sure, in the last slide, are they to see what is the best process to use, and is it available for everyone?
Yeah, so it's not available for everyone yet, but we are in the final stages of writing up a paper about this system. So if all goes well, it should be submitted before the end of the year. And in the process of submitting, we're planning on pre-printing it, which will also make the code available. And yeah, the main direction it's aimed at the moment is to interactively determine what the best pipeline would be. You can use it is this designed fully featured that you can actually use it for proper analysis and export all of the CSV files that you might want and export images and everything. It has that capability, but in my personal opinion, it's more helpful as a tool to try various versions of pipelines and then tweak the parameters. Like, okay, if I add this much blur, what is my segmentation look like? If I add a little bit more blur, is it better or worse? If I do some gamma correction, where should the best value be? That interactive ability of it is immensely helpful to just speed up the process of determining what the pre-processing pipeline is, especially for more complicated images. For simple things, you don't really need it, but for complicated things such as small structures and specific structures like mitochondria or autophagosomes or lysosomes or these types of things, it's been really helpful just to speed up the process of coming up with the pipeline to start with, and then you can entrench the pipeline more in a script that you then do more batch processing with.
Thank you for that answer. And so maybe as a closing, are there specific for an image analyst who's starting out on their journey and is interested in seeing what more they can do with machine learning and AI? What would you recommend as sort of the starter tools, the starter pack in terms of software that you would recommend?
Yeah, so I think the natural journey would be to start with just recording macros in Fiji and then seeing just what would be sitting behind it. There are some basic machine learning plugins in Fiji like Stardust and stuff like that, which is already able to do some segmentation and automatic minorization for you, improve watersheding and stuff like that. So just looking at the plugins that are available in Fiji, getting used to recording macros, setting up some automatic pipelines, just getting comfortable with the code. But at some point, I think the jump will need to be made to something like Python. As soon as you switch to Python, you have more access to things like the Pori, which is a great image processing tool. Just knowing how to use skimage, even the entire machine learning ecosystem of TensorFlow, PyTorch, all of these things are running primarily in Python. So learning Python as a base, and then immediately coming up with some simple problems to solve, and then trying to learn the tool in the process of solving that. I found that's the best way to motivate you, is to have an actual goal in mind instead of just working through tutorials and then learning the tools that's necessary to achieve that goal, because there's just too much to know and you can't possibly learn everything before you even start tackling problems. So get a bit of a base. Like I said, Fiji is a good base, but then just tackle an actual problem in Python and then learn as you go.
Giving yourself small digestible problems to deal with up until you build a skill set to deal with the bigger picture. That overwhelming.
That's a good way of putting it, yeah.
All right, I see Ali, your hand is up. Please unmute and ask.
Yeah, thank you. Thank you so much for the talk. That was very valuable. I just have a question regarding the MELPG plugin. I see that it's able to pinpoint fusion and fission events. I wonder if this could be applied not only to mitochondrial images, but if I give it, like, for example, images of an immune cell and a target cell, and I want to see events where they get adjacent. Would this plugin work for such scenario?
Yeah, it's definitely developed to be very general. It's not like we've applied it in the context of mitochondria just because mitochondria is a very hot topic, and it is quite a challenging use case for it. So that's kind of what it's positioned as, but it's very general. If actually, I don't know exactly about your use case. I would have to look at the images, what they look like, to say whether it will be exactly transferable or not. But we've applied it to something like peroxisomes, where we weren't sure if peroxisomes are fusing or not, or whether they just move close together. And we wanted to automatically pick up where these potential fusions happen. And instead of trying to go through hundreds of images by eye and trying to pinpoint that, we just pushed it through this mouse system, and then we got all of the event locations automatically, and then we could just focus our attention on those locations. So yeah, it's definitely developed to be general. I'm not tied to mitochondria in any way.
Perfect. Thank you very much.
Cool.
Thank you. So if there aren't any other questions, if we do have questions, please feel free to put them in the, or raise your hand if you'd like to unmute. I'll give you maybe a minute or two. The questions. Rensu, thank you so much for this presentation. This was a very, very interesting and informative, and hopefully it has kind of shaped our perspective of how we can use AI, and kind of maybe has piqued an interest in others who are trying to find solutions to problems that they've had and ways to answer questions. Can people contact you should they have any further questions?
Yeah, you're welcome to. I'll maybe put my, there's a little website that I have. You can find my contact details there, or I'll be putting my email address also just in the chat if you want to email me directly.
Thank you very much. So everyone, please do look in the chat for contact details on how to get a hold of Rensu. If you'd like to see this recording, it will be put up next week on the ABIG YouTube channel. And please do continue to follow our social media for these and our newsletter as well through our website to find out more about these webinars and more tools and sources that can help you with your imaging and your analysis. And with that, Rensu, thank you so much. Thank you all for attending, and enjoy the rest of your afternoon.
Yeah, thank you. And thank you, Precious, also for chairing this meeting.
Sure. Everyone.